Hello, everyone. Thank you so much for tuning into my virtual talk. I wish I could see you and that we could have a cup of tea together and have a chat after this, but please don't let this virtual barrier stop you from emailing me afterwards if you have any questions. Always happy to chat. Today, I'm going to be sharing about some of our latest work regarding the imaging of inflammation and different aspects of the immune response in the context of brain diseases. And so why do I care so much about imaging immune responses in the brain? Well, over the last decade or so, in particular, neuroinflammation has emerged as a key pathological feature of pretty much every disease that affects the brain, whether it be a neurological sort of neurodegenerative disease, or whether it be a psychiatric illness or infection-based disease. An example of the latter would be long COVID, which we've heard a lot about recently. So when there's an infection peripherally that can end up having an impact on the brain. And so what is it? What is neuroinflammation? Well, it's any inflammatory process that is present in the central nervous system, typically involves microglia, astrocytes, oftentimes peripheral immune cells that have been recruited to the brain, and it leads to the production of cytokines and chemokines, these chemical messages. It's usually transient and beneficial, leading to the mitigation of pathogens or the clearance of damaged tissue, so on and so forth. But when inflammation in the brain is not resolved, when it really persists, this is when it can become particularly problematic and can lead to cellular dysfunction and neuronal cell death, which is what we see in diseases like Alzheimer's, unfortunately. So it is this really common underlying feature in many brain diseases, as I just mentioned. And I strongly believe because of that, it really deserves our attention, and therefore we need suitable tools to be able to study this phenomena. And that's where I think PET imaging can play a really big role and can be very powerful in its ability to shed light on the role of different aspects of the immune response in these diseases. And so along that along those lines, we've been doing quite a lot in this area. My lab has been developing a number of PET imaging approaches to better understand neuroinflammation so that we can better modulate it and treat diseases such as ones that I just mentioned. And so I'm just going to show you a few examples of some of the work we've been doing, and then I'll go into detail in for a few of those examples. So firstly, we're currently really delving into the area of imaging the whole body and brain in a number of diseases. This is an example of where we're looking at inflammation that occurs in ME/CFS, which is chronic fatigue syndrome, also in long COVID, so really looking at peripheral infection and how inflammation can be fueling a disease that's occurring in the whole body and brain. And here's some example images of where we've used TSPO PET to visualize this kind of inflammation, and we're doing that at Stanford at the moment. Another thing that I'm really excited about in the lab is that we've developed this strategy for really delineating peripheral infiltrating immune cells, really peripheral infiltrating innate immune cells from those that are present normally in the brain. And we can do this because we discovered a biomarker called TREM1 that is only on these cells. And we made a PET tracer for it, and here's some example images of what we've been doing in the realm of imaging innate immune activation for multiple sclerosis. And I won't have time to go into this today, but it is a fascinating project that I'd love to talk more about if anyone is interested. Another area that we've been fascinated by and working on in the lab is imaging and adaptive immune cells, so looking at your B cells in the context of multiple sclerosis. And I will expand on this a little bit later in the talk, but we've developed tools to look at both CD19 positive and CD20 positive B cells. A project that was started by Dr. Sam Gambier that I'm now working with some members of his former lab to continue is looking at T-cell activation markers in cancer, and in particular in the context of CAR T therapy, and also sort of looking at these in CNS diseases, because that's my personal interest. But we're really pushing forward a number of these agents that target OX40 and ICOS, some really promising biomarkers, so watch that space. We've been using a number of high throughput gene expression assays to streamline the identification of promising biomarkers of toxic inflammation, and this could be a whole other talk. I'm excited to say that we've identified a number of really promising targets using this approach, sort of looking at tissue from both mouse models and humans. It's post-mortem tissue from various diseases. We've also been looking at Parkinson's disease, working with Dr. Thomas Montine, the chair of pathology at Stanford, looking at imaging what's going on in the periphery and the spleen, and what's sort of trafficking up the vagus nerve, for instance, and into the brain. And we've been doing that with TSPO and TREM1 PET. And we've also been looking at developing a number of very new imaging agents for visualizing activated microglia and macrophages, so something that's sort of beyond TSPO work, where we've been looking at some of the targets that we've been discovering in number 5 here and making imaging agents. And I will be expanding on one of our newest agents that actually has no target or no receptor that it's targeting. That'll be one of the first projects I talk about. And last but not least, we've been investing a lot of time into developing new in silico tools for predicting the success of PET tracers for imaging the brain. So instead of trying to talk about all of these projects, as I said, I'm going to share about 3 intriguing questions that we've been working towards answering in the lab that are related to these project areas. So question number 1, can we develop a PET tracer for imaging activated myeloid cells without targeting a receptor? I think we can, but how do we do it? Well, we started a collaboration with Ashvatha Therapeutics, and we identified OP801 as a candidate agent to radiolabel. It's actually a dendrimer, so it's shown here. It's one of these. It's a 4th generation hydroxyl PAM-MAM dendrimer that's made of repetitive branching units of methyl acrylate and ethylene diamine. Has a molecular weight of about 1500 Dalton. And so why dendrimers? Well, instead of having to target a specific gene or protein receptor, OP801 and dendrimers like it are able to cross the blood-brain barrier in the presence of neuroinflammation and are specifically taken up by activated microglia and macrophages throughout the body and brain. And this has been shown in a number of different models. So really the specificity of this agent in particular that I'm showing for activated myeloid lineage cells has been demonstrated through over 30 animal models that have been published in the literature, including dogs, non-human primates, mouse models, and here I'm showing you an example of some staining that's been done after the fact. Well, actually, this is staining after injecting a 5x FAD mouse model. So an Alzheimer's mouse model has been injected with a fluorescent version of this dendrimer. And what we can see is that the dendrimer is shown here in red, and we can see IBA1 is highlighted in green. So you can see the overlay of the red dendrimer and IBA1 highlighting all of the activated myeloid lineage cells, and amyloid is in blue. And time again, what has been shown with this particular dendrimer is that it is taken up specifically just into the myeloid cells that are activated and angry. And I didn't believe it until I saw it, but this dendrimer does indeed get across the blood-brain barrier and is only taken up into those cells. The mechanism is fluid phase endocytosis, and the half-life is relatively short. We were tasked with figuring out a way to radio label this in a robust and reliable way. And I want to give a shout out to Dr. Bin Shen, who's the director at the cyclotron and radiochemistry facility at Stanford, and my grad student Isaac Jackson. They've both been working really hard on this synthesis and really have developed a way to make this in a streamlined manner, such that it's usable for routine clinical production. And so that work actually will be presented at WMIC this year, and there'll be a paper published on the routine clinical production path, but we also have another paper that just got accepted into molecular imaging and biology on this agent, so keep a lookout for that. But we were able to make this agent via a two-step radiochemical reaction. And basically what we do is, firstly, we prepare the fluoropropyl azide from its tosylated precursor, and then we use we take that molecule and we perform click chemistry. So then what we do is we take our alkyne functionalized dendrimer precursor and react it with the fluoropropyl azide in methanol. And here we're showing that we have some optimized conditions now. These have been optimized even further, in fact, just as of last week. And the very exciting news is that all of this chemistry has been worked out to the point where we've submitted a DMF and an IND, and think everything should be approved by the FDA shortly. Okay, so after making the radiotracer, we injected it into healthy mice, and my other grad student, Mackenzie Carlson, spearheaded all of the in vivo work. She did a fantastic job. Here, what you can see is that there's only uptake or a signal in the kidneys and in the bladder. So it's really just being excreted when in a healthy setting. So meaning that when there is no activated microglia or macrophages around, this entity is just goes into the bloodstream and is eliminated very rapidly. And that it does happen very, very quickly. And you do not see signal anywhere else. And here we're just showing some of the HPLC curves for our final product. And then we also measured the stability of this agent ex vivo by taking blood from the mice that have been injected at various time points after injection. This is actually at 150 minutes post-injection, which is a very late time point. We actually only perform our imaging studies up to about 60 minutes or 90 at the most, but there's only one radioactive peak present at that time, which is excellent. That's exactly what we had hoped for. And here we are. We have our first imaging study with this agent in mice, and we use just a classic LPS model. Just by injecting 10 mg per kg of LPS intraperitoneally. And we did that, and after injecting it, we actually waited 24 hours, and then we injected our radiotracer, OP801, and then we performed dynamic 60-minute dynamic PET-CT imaging. And here are the images. This is our saline injected control. Looks very much just like the untouched healthy control you just saw. And then we have our LPS mouse shown here, which looks quite different to the saline control. You can see a lot more signal in the blood pool, in the abdominal cavity, the spleen, liver, all areas where we know there's a lot of inflammation occurring in this model. And then if we zoom in on the brain, you can see in your saline control where there's really no signal at all, very negligible. And in fact, when I first saw these images, just the saline control by itself, I thought, did this mouse get injected? Because we were just looking at the brain and there was really nothing there, which is quite remarkable, because with most PET tracers you see uptake around the salivary glands, in the Hydarian glands or other parts of the snout, but here you just see nothing unless there is active inflammation occurring. And so nothing in the saline control, And then in the mice that have a sort of moderate sepsis score, so we score them using this murine sepsis scoring system, which looks at activity of the mouse, sort of its grooming behavior and a whole set of features. But moderately scoring mice, moderately sick mice, had quite a bit of uptake occurring in the brain. And then as soon as you see the severe, or we image the severe scoring mice, you can see the signal is markedly increased. And in fact, what I find really powerful is that our in vivo sort of whole brain signal really does correlate very nicely to the murine sepsis score, as shown here in this graph. So that was very compelling to me. And then after using that very simple model, we went to a little bit more of a sophisticated model using a 5xFAD mouse, so a mouse model of Alzheimer's disease. And we wanted to image very early on. So these mice are 3.75 months old. This is at a point where there are certainly some plaques and inflammation, but really it's only starting at around 2 to 3 months that those plaques begin to form and the inflammation. So quite an early stage. To put it in perspective, most studies using these mice where there is imaging, people look at about 6 months. That's the time point people usually image at for TSPO agents in this model, for instance. In any case, we wanted to see how early can we detect inflammation. And you can see from these rotating images that it's quite apparent that there is an elevation in signal in the transgenic mice shown here compared to signal in the wild type mice. So that was very exciting, and we were absolutely thrilled to see this. Then we compared in the exact same mice imaging with a TSPO PET agent here, GE180, which is an excellent radiotracer for rodents, not great for humans, but excellent for imaging TSPO in rodents. Unfortunately, we couldn't really tell the difference between the transgenic and the wild type mouse at this time point. We know at 6 months we start to see very robust differences with GE180 or other TSPO tracers. And here's just sort of breaking down the numbers for you that are directly corresponding with the pictures that I just showed. So OP801, our new dendrimer imaging agent, you can see the ratio of transgenic uptake in the cortex to wild type uptake in the cortex. It's about 3-fold higher uptake for the transgenics, and it's about 3-fold higher in the hippocampus and whole brain. Whereas for TSPO, there's about a 10 to 20% increase, and that's what we used to get excited about or thought, oh, that's something's happening. But it's not quite sensitive enough, and that's what we're concluding at this point. And where at the moment we're actually repeating a lot of these studies and expanding the numbers. Our take home for this question is that OP801 enables sensitive visualization of endocytosing microglia and macrophages in the whole body and brain of different models that whereby inflammation is occurring. So we're really able to visualize that process, which is absolutely exciting in my mind. And next steps, we're going to be completing the 5xFAD studies. We have a number of mice that are sitting at the moment, aging, and we'll be imaging them in the coming months. And we're also looking at depleting microglia in this model so we can assess specificity. We'll also be investigating which myeloid subsets in particular are predominantly taking up this tracer in different disease contexts and also after different treatments. So for instance, we're looking at EAE at the moment and we're treating with various drugs. So we're going to be doing RNA-seq and a whole range of studies to really understand what the image truly represents in terms of the myeloid cell subsets. And then most excitingly, we have clinical studies that are starting, and in collaboration with Andre and his team, Farshad Moradi, Dr. Farshad Moradi will be heading up these trials, and we'll be looking at healthy controls and ALS patients first, followed by Alzheimer's disease. So question number 2, something that is very close to my heart is multiple sclerosis and trying to better treat it. the really nice thing, I guess, if there's something nice, but maybe encouraging thing about multiple sclerosis research is that there are over a dozen approved disease-modifying therapies for multiple sclerosis. So that's exciting, because actually for pretty much every other brain disease, or most brain diseases that are that have degeneration and extensive inflammation, we don't have those sorts of therapies. But for MS, we do, and that's great. However, not every patient responds to a therapy that they're put on. Why is that? Well, and it could be for a number of reasons, but the bottom line is that MS is very heterogeneous. People can have quite different immune signatures, and it is really an immune-driven disease. So it's important for us to understand which immune cells are playing a more prominent role for a particular patient, so we can decide on the therapies and sort of get them onto a good management plan. So I've had this question in my mind for a while now. Why don't approved disease-modifying therapies work for all patients? Or how do we select the best therapies for an individual? And one of the cell types or types of therapies that really piqued my interest is therapies that target B cells. And I became really interested in this after hearing about the dramatic response that a subset of MS patients have had to anti-B cell therapies. But I learned that there was a variable response, that not all people responded. And that has a lot to do with the number of B cells that one might have in their brain. We know that and beyond the brain as well, we know that there are these B cell follicle-like structures that you can find in the meninges, in the subarachnoid space, in the actual parenchyma as well. And we also know from post-mortem studies that some people can have just a few of these follicle B cell clusters, or follicle-like B cell clusters, and others may have 10 or 50, and some people might have hundreds. And they're really the people that we want to find and put on these sorts of therapies, because we believe they have a more B cell driven disease and could really benefit from having a therapy that will knock out a lot of the B cells. And actually, ocrelizumab, which is an anti-CD20 sort of anti-B cell therapy, is the only therapy that is approved for primary progressive MS, and which is great because we don't have any other therapies. But why all patients don't respond is still a question, and maybe it has something to do with the delivery of the medication into the brain. Maybe it's a dosing issue, but really the standard of care right now is just to treat people and then to measure just via a blood sample how many B cells are being depleted peripherally in the blood. And you may see complete depletion in the blood and think, great, we've got the right dose, everything's being depleted, but that doesn't necessarily tell you what's happening in the brain. And so I thought that it would be a good idea to try to visualize that using PET. And so we thought, which kind of a marker should we or could we use to target a PET tracer to? We have, myself in collaboration with Dr. Sam Gambier, we made an imaging agent targeting CD20, and that was rituximab. And we have published on that and have some excellent and very promising preliminary results on that really motivated myself in my lab to continue working on imaging B cells. But what we found with CD20 imaging is that we were missing some of the key B cell subsets. So plasmablasts and plasma cells don't have CD20 expression. And those cells are actually the ones that are believed to be causing a lot of trouble in the brain. For instance, they're secreting antibodies. And folk are really trying to target these cells now for therapy. And there's a number of anti-CD19 therapies being pursued. So I thought if we want to capture more of that B cell pool, it would be beneficial to try to image CD19 positive B cells. And so that's what we did. And Carmen Azevedo in my lab, a very talented radiochemist that's just gone into her grad school at WashU, very exciting. She developed a CD19 PET tracer. So we initiated a collaboration with Viela Bio. We labeled one of their control antibodies. So they developed a therapeutic antibody for their trials, but we didn't want an antibody that killed B cells. We wanted an antibody that could be a quiet observer that would allow us just to visualize and track these cells. So we used a mutated variant of their therapeutic drug, their therapeutic antibody. It's known as 16C4TM for short. It lacks the ability to elicit B-cell depletion via antibody-dependent cellular cytotoxicity due to mutations in the FcR gamma region, while maintaining affinity for CD19. So perfect imaging agent. We just did some very simple DOTA conjugation and labeling with copper-64. We've also made a Fab of this and labeled it with zirconium. We're going to be labeling the MAB with zirconium as well to allow us to track. But this was some of our initial work. And what Carmen found was that we could really visualize very clearly and with a lot more clarity than rituximab, for instance. We could visualize B cells in the white matter, the brainstem, meninges. We also see more B cells in the ventricles compared to a naive mouse. This is an EAE versus naive. And Samantha Reyes in my lab, who just got into a PhD program at Stanford, very excited for her, just has been doing some therapy monitoring studies using this agent and this mouse model. So let's talk a little bit about that. I only have a couple of slides on that, and then I will move on to my last area to talk about. But this is a very interesting project, I think, and it has a lot of potential to have impact in the clinic. So here what we're doing is we're assessing the ability of this new imaging agent, this CD19 MAB, anti-CD19 MAB. It's actually very specific just for the human CD19. So we're assessing the ability of this agent to detect B-cell depletion in the CNS and peripheral tissues of EAE mice. Now, these EAE mice are human CD19 transgenic mice, so they have human CD19 expressed on all of their B cells, all the B cells that are supposed to express CD19. And we're looking at imaging these mice before and after an anti-CD19 therapy. So, sort of what we would want to do in the clinical setting. And so, what we do is we start by inducing EAE. And then what we do is we administer the therapy at about day 7, so very early stage when we just start seeing a symptom of the disease. So just, it's like the initial signs. It's like very, I think, translational, or it aligns with what you would see in the clinic with somebody coming in when they have their first sign or symptom. And so we administer that drug. And then we image a week later and see what we see. And we do not only imaging, but also some flow cytometry, some ex vivo gamma counting, autorad, immunohistochemistry, a little bit of everything. So we're trying to get everything we can from this experiment. And in imaging a lot of EAE mice, we discovered that because this is a very spinal cord focused model, in the sense that a lot of the pathology is occurring in the spinal cord, but also the brain. What we discovered is if we position the mice on their backs, we can stabilize the spinal cord and the brain and really minimize motion, because that's where we want to be drawing our region of interest. That's where we want to be quantifying our PET signal. So if we have them all on their backs and tape them down securely and pop them into the scanner, and we're imaging 4 at a time here, we get really high quality images. And we've done a direct comparison between sort of mice that are prone versus mice that are supine. And just wanted to share that in case anyone else in the audience is imaging EAE mice or any brain imaging in general. I think this ends up leading to higher quality images. And so what were our results? Well, here we are. This is an image of let's see if I can get that. Yes, here we are. This is an untreated EAE mouse. And what we see is a lot of signal in the spleen, which is where there are a lot of B cells, so no surprises there. Also in the lymph nodes, where there are B cells, and in the bone marrow, where we find certain subsets of B cells as well that express CD19. So really lighting up the areas that we would expect for a tracer like this. So really beautiful. And then in a mouse that has been treated with CD19, we see the spleen signal has gone away. The bone marrow signal is very low, negligible. You don't see the lymph nodes anymore. So you think, oh, this is great. Looks like the therapy worked. Fantastic, we're done here. No, unfortunately, no, that's not the case, because this mouse here is still rather sick. Some mice got better, but a number of them did not. And that's a problem. And that's actually what we see in the clinic. This mouse did not get better. It still had paralyzed legs, unfortunately. So what was happening? Well, we looked at the brain, we quantified the signal in the brain, and what we could see from this is that we had a reduction in the signal from the untreated to the treated, but it wasn't fully reduced. There's still signal there, and there was some variability, but it seems that for the most part that signal is just still there, and not fully decreased. So what does that mean? Well, we took all the tissues out of the mice and measured them in the gamma counter just to get a better look and better resolution on this. And what we found was there was still a lot of signal. Well, we didn't get any differences in the brain here. There was nothing in the spleen, still some in the bone marrow as well. And it seems that we still had signal in the CNS tissues. So then we looked to flow cytometry, and what we found was while we had complete depletion in the spleen of B cells, of the CD19 cells, we had only partial depletion of CD20 and CD19 positive B cells in the brain and in the spinal cord. And so that tells us that maybe we're not depleting all the B cells and the ones that are causing the problem. And then we did some staining. And what we found in the untreated was, yes, beautiful B cells in the meninges, parenchyma, and the ventricles, like we would expect. And in the treated mice, while we saw a big decrease, actually almost completely complete depletion in the meninges, we still saw B cells in the parenchyma and some in the ventricles. So this told us that we weren't able to quite deliver the treatment to where it needed to go, or it wasn't able to access all of the B cells. And maybe we need a different strategy there, or perhaps a higher dose, or more continuous dosing. In any case, this did shed some light on why that mouse, or why some of the mice were still sick. And so we're excited about that, because it seems that we have an imaging approach that really allows us to shed light on the whole body and brain sort of adaptive immune response in the context of EAE and hopefully MS. So what we're doing is we're assessing this in other EAE models at the moment. We have a new model that we're working with a lovely collaborator at the University of Iowa on. We're working with Genentech using some of their anti-CD20 therapies, and we're optimizing the radiolabeling to translate this to the clinic so we can start using this in the context that we really want to be using it in patients. Okay, in the last 10 minutes or so, I'm going to be sharing about something that has been really intriguing me for quite some time now, and motivating me to think about different solutions, and that's the issue of getting small molecule radiotracers across the blood-brain barrier. It's difficult. It's not straightforward, and actually less than 90% of small molecules, and basically all large molecules, can't pass the blood-brain barrier, or greater than 90%, sorry. Most agents do not cross the blood-brain barrier, and that's tough. And so I had this dream, or I've had this dream for a long time now, of having some kind of an app on my iPhone that I could just take a photo of a molecule that I was interested in, and for that app to tell me yes or no, make this agent or not, because this will cross the blood-brain barrier, this one won't. Perhaps if it says no, it could give a suggestion as to how to cross the blood-brain barrier if you modify this group or that group. So I've had this idea of developing a way to predict whether a molecule will cross, and sort of getting guidance based on an app of some sort. And I think some of my grad students thought I was crazy. But, during the pandemic, we had a little bit more time to do crazy things and think about all sorts of wonderful new techniques. And we started working in this area. So this is just to remind you all of the general workflow for small molecule CNS PET tracer development. It's quite long and arduous. We start with biomarker selection and target validation, and then we move, once we have a great target, we move to designing or engineering a tracer. Organic synthesis typically is what is needed at that point, and that can take a long time. It's usually something like a 4-step or a 9-step reaction to get to your precursor, and another few steps to get you to your standard, or the other way around. And then you need to do your in vitro characterization, typically in cells. And then if it doesn't look good, you start optimizing, and maybe you'll start again if you're not getting binding. And then also maybe you'll think about ways to develop the radiosynthetic method and do some preclinical imaging. And if that doesn't look good, you might go back to the drawing board. If it does, you may get to clinical translation. So it's an iterative process, and it's not easy when it comes to the brain, because not everything you think will cross, and then you're stuck. And really, predicting CNS uptake and non-specific binding of tracers prior to the in vivo testing stage remains a significant challenge. And it's really quite resource intensive, as you can imagine, especially for a small academic lab. So there really is this unmet need for in silico tools in particular that are tailored to PET tracer designs that can help us to decide and prioritize particular molecules that have a higher likelihood of success. And so if we had such tools, they could have a massive impact, I think. I would love to have such a tool, and that's what's been motivating us. And I know a lot of our collaborators would too. It really could have a big impact on increasing or facilitating the translation of traces that can be used for the CNS in more of a time and cost-effective manner. My 2 students that have been working hard on this are Isaac Jackson and Audrey Liu. They've been really doing an absolutely wonderful job on this. I'm so proud of them and excited to tell you about this. So what we have here is we have the some examples, papers of where people have used in silico tools for drug design. That's this first paper, and used something called MPO, or this multi-parametric optimization sort of algorithms and ways of prioritizing molecules that could cross the blood-brain barrier. Unfortunately, this is really focused more on therapeutic drugs, not on tracers, although people use this tool to predict whether a tracer molecule would get across the blood-brain barrier. And you might think drugs and tracers are similar, but actually they're not. And they're really not at all. I mean, some similarities, but there's a lot of differences. And then some folk, or at least this paper in particular, but people have been thinking about how do we develop modified versions of this for tracers, and there's been some papers published. Unfortunately, the libraries for these tools are not great, meaning that the library of molecules that they're drawing upon to learn what properties are important are small. And actually, every single negative control for this paper is a non-specific binding molecules. They still all cross the blood-brain barrier. And so, it's a start, but not the most ideal from my perspective. But that's what we had. And so, while we were developing our tool, we were trying to develop some new molecules for imaging microglia. And we wanted something for P2Y12, which is an interesting target. And we searched the literature for all the different molecules available. We picked this one out because we were looking at the properties of all the available molecules that hadn't been labeled yet. And we thought this one looks reasonable. And when we checked via the CNS MPO, so that first tool I was telling you about, it got a good score. Actually, anything over 3 predicts that the molecule should cross. And we use the PET MPO. Again, anything over 3 is a good score. So we're getting 4. or 4 or over for both of these. So we thought, okay, well, maybe we'll give this a go. We did. Unfortunately, you can see a big black hole in this monkey image, which is what happened when we imaged both mice and monkeys. So very sadly, we did not predict with the current tools that this tracer wouldn't cross. We thought that it had a chance. So back to the drawing board, and actually this motivated us even more to make our own tool. So here I'm showing you parts of the library of molecules that we put together. So we collected a number of positive controls, negative controls. In fact, our library we have the most positives and negatives that anyone has compiled so far for pet tracers. 75 positive controls. They're all verified in humans to be successful tracers. We took a lot of care to include diverse negatives. And what we did is we had non-permeable molecules, molecules that are high non-specific binders, ones that are substrates for PGP, so they're actively pumped out of the brain. We're still building on this, so if anyone has any negative controls for us, please let us know. If you've made a molecule that didn't cross the BBB and you'd hoped it would, maybe it could help us and the whole community. We've also been looking at a number of different parameters associated with all of our molecules to draw upon as much information as we can. And here what we're doing is we're looking at all the different properties, and here are just some of the key ones we've been looking at. We found that if you compare the successful to the non-permeable CNS PET tracers, something that sets them apart is the C-log P, and surprisingly also the polar surface area, which is really interesting. And when we manually assign things, we're actually quite good at it. And then we actually use some AI. We use a support vector classifier, and we're even better at differentiating positives from negatives. And this is way better than what's available right now. So we were super pumped about that. We tried to differentiate non-specific binders, so tracers that have failed because they have high non-specific binding, from the positives, but we couldn't. We're not very good at that yet, but that's okay. We're going back to the drawing board on that. But we are really good, I think, at predicting whether something will cross the BBB or not now with our new tool. And so I think we're ready to answer this question. And we indeed put our new tool, to good use when picking a molecule for this new target, MGX, to look at microglial activation. There were over 500 molecules in the patent literature, and we ranked them all. We ran them through our new algorithm, and we came up with one molecule in particular that had these properties. And this is the score for the CNS MPO tool. This is the score for the PET MPO tool. So you think, well, these are really great scores anyway. You would have picked them with the old tool. This is our new tool, and you might think 3.13 out of 5 seems a bit low, but actually our scoring system is out of 5, and anything between 3 and 3.2 gives us a 95% positive predictive value. So we were very encouraged by this, and we imaged a monkey, and lo and behold, we get a beautiful brain image. So excited about that. And then what I think is most powerful is we went back and ran our old molecule, the one I first showed you, through our new tool, and it got a score of 1.66 out of 5, and that we would have never picked. Anything less than a 2 you would not pick using our new tool. So we never would have made that mistake. So hindsight is 2020. We've validated our own tool. We're actually validating it more at the moment. We're expanding upon all the different physicochemical properties we're looking at. We're looking at an additional greater than 70 descriptors, and we are making a second generation tool at the moment. We're working with some folk on developing the app for this, and looking at a whole range of other machine learning methods, and comparing our sort of running different libraries and test sets through our tool. So in summary, here we are. This is what I've talked to you about today. We have a new imaging agent for microglia and macrophages that's a dendrimer that we're putting into people this year, so stay tuned for that. We have a very promising way of tracking CD19 positive B cells in MS that we're hopefully going to be doing more therapy monitoring studies for in the near future. And we have this new in silico tool that appears to be able to predict BBB penetrance for candidate traces. And so we're going to be doing a lot more work this year. We're busy working away. Hopefully we'll see you at some conferences later this year. This has been a huge team effort. Everyone on this page has helped so much. And I'm just so thankful for them all. And I'd love to hear any questions. If you have any, please do let me know. Okay, thank you so much. Enjoy the rest of the meeting. All right, bye-bye.